Okay, I'm just getting the setup going. Let me stop the video, share the screen, and get my PowerPoint slides going. So now we're on to the section on esters. It starts off with preparation of esters, and I'm going to combine this. We got section 20.10 and 20.11. I'm going to combine the two of them together for this video. We'll see how it goes. So preparation of esters. The first way we can prepare esters works pretty well for certain relatively simple groups, but we can take our carboxylic acid and treat it with sodium hydroxide, and as we saw in the previous video, that generates the carboxylate anion, and that's a good nucleophile. And what we have here is just a simple SN2 reaction, and then that can kick out the leaving group here. So in this case, it's just metal iodide. Iodide gets kicked out, and it's very good for making metal esters. For more complicated ones, we could use one that we saw in the acid chlorides, where you take the acid chloride and react it with your desired alcohol in the presence of pyridine. But the classic reaction of esters is this one coming up next, which is the Fischer esterification. So this is where you take an acid and an alcohol and you get an ester and water. So we have acid plus alcohol produces an ester and water. And notice we have equilibrium arrows here. It's catalyzed by acid, but this would be a case where we would maybe use something like our Dean-Stark apparatus that we talked about in the chapter on aldehydes and ketones when we were making things like acetals. We would use that Dean-Stark apparatus to remove water so that we can shift our equilibrium to the product side. So again, when we're thinking about our reaction here, it's a nucleophilic acyl substitution reaction. We got our core steps, right? We have nucleophilic attack. We have loss of to form that tetrahedral intermediate, and we've got loss of a leaving group, and we've got all those different places where those proton transfers can occur. So let's look at our first step in this. I'll move to a fresh slide here. So our first step in the case of the Fischer esterification is a proton transfer. So we're going to have methanol as our alcohol here. We're under acidic conditions, but we're not under acidic aqueous conditions. Having any water around here, the mechanism won't go to, the reaction won't go to completion. So our first step is protonation of the oxygen of the carbonyl. That, of course, gives us this species, which is now more electrophilic. I'll draw out this resonance structure just to remind you of one of the reasons why it's more electrophilic. You got this resonance structure here, and now we've got a positive charge there. But having that positive charge on the oxygen after that protonation makes that carbonyl carbon more electrophilic. And then we get nucleophilic attack. So the methanol can act as a nucleophile. We get our nucleophilic attack at our electrophilic carbonyl carbon. We push the electron density up onto the oxygen. And we have our tetrahedral intermediate. And then at this stage, we could reform the carbonyl and kick out the methanol, which is probably occurring. But we need to turn one of those OH groups into a good leaving group. So it's just a series of proton transfers. So we remove the positive charge from the oxygen of the methanol species that acted as the nucleophile. Should be a bit more precise with my curved arrow there. Then we We've got this intermediate here, but we're going to protonate on that hydroxy group. So we get proton transfer. Let me correct that little glitch there. It happens from time to time. So now we're set up with our good leaving group. And so we can reform the carbonyl and kick out the leaving group. Leaves us water. And then the last step is another deprotonation. So methanol, another molecule of methanol, or the water we just lost can act as a base. And then that's another one of those weird glitches. Not sure what's going on today, but let me clean that up a bit. And then we get our desired ester at the end. So quite a lot of steps here, but really, let me change color here and I just highlight these. Come on. We have a proton transfer. We have a nucleophilic attack. We have a proton transfer, loss of leaving group, and then a final proton transfer. Again, just some of those core steps that we learned about way back in Org 1. Now, the reason why this mechanism is like this, let me go back a couple slides and just get a clean slide here. This has been proved by some isotopic labeling. So let me just show you this. If we take an ester, take a carboxylic acid, and we make or have methanol that has a heavier isotope of oxygen, and so this, we often use oxygen 18, and look at where that ends up. We see that oxygen is this oxygen in the ester product. And that kind of rules out some of the other possibilities where you might think it could be something like an SN2 reaction where the OH of the carboxylic acid is doing an SN2 reaction on here if this has been turned into a good leaving group. So another one of those ways to test for mechanisms. So let's move on and look at some reactions of esters. And this one is, I guess, kind of timely. It's the reaction that's used to make soap. Soap, of course, being one of our main ways to help prevent the spread of COVID-19. And it's a reaction that's been known for a long time. The way the saponification reaction works is it's essentially a basic hydrolysis of the ester to give us a carboxylic acid. So we still kind of call it a saponification of an ester. And I'll just do the mechanism very quickly here, and then we'll take a look at our example of making a soap. So in this case, we have our ester, we have sodium hydroxide. You might have noticed by now that any of the reactions we have where the nucleophile has a negative charge, the reaction mechanisms tend to be more straightforward. We get a nucleophilic attack at the electrophilic carbonyl carbon. Our nucleophile is good enough that we don't need to. So let me correct that little glitch. Oops. Our nucleophile is good enough that we don't need to have any proton transfers occur before the nucleophilic attack. So we get that tetrahedral intermediate again. We have our carboxylic acid. We reform the carbonyl and kick out the leaving group. And when you initially look at this, you might be tempted to stop at this stage, but actually it's equilibrium until this point. But we've generated an alkoxide, which is a pretty strong base, and the reaction is driven to completion by this deprotonation, and it produces the carboxylate initially. And so if we were doing this in the lab and we wanted to generate a carboxylic acid, we would follow that up with the dilute aqueous acid workup so that our carboxylic acid will get protonated. But without this deprotonation step, the reaction doesn't go to completion. And remember, we're talking about a carboxylic acid that has a pKa of about 5, and the alcohol has a pKa of about 16. So you can see that this conjugate base here will do that deprotonation. So let's take a look at this example in the bottom left-hand corner of the slide. We've got a fat molecule, so that consists of a glycerol part. Let me just change my ink color here, which is this part here, linked to different fatty acid molecules. So fatty acids are the protonated versions of this. Again, I got that weird little glitchy bit there. Really don't like it there, so let's try that again. So when that carboxylate has a proton on there, that's a fatty acid. The sodium ion will be gone. That would actually sorry, let me change my train of thought there. Those fatty acids, there's 3 fatty acids to each glycerol molecule, as you can see. And when we do the saponification reaction, we generate the sodium salts of the fatty acids, and that's what acts as the soap. So we have a molecule that has draw it over here. That's not great, but has a hydrophilic head and a hydrophobic tail. And so that can act as a soap. So typically your dirt would be hydrophobic, and it will in water these form kind of micelles around other hydrophobic species. So let me get a micelle being drawn here, and so on. We'll complete the rest of that. And of course, your cell membranes are made up of compounds that have some triglyceride properties, usually, of course, more complex head group than a glycerol, but essentially the same idea. And so soaps can kill bacteria and viruses by breaking those membranes apart, by interfering with the membrane structure. That's a very basic explanation of it. Okay, so that's the saponification, relatively straightforward mechanism, but don't forget that you need this step here in order to drive the reaction to completion. Let's now look at the acid hydrolysis of an ester. So have a guess what the first step of this mechanism is. We're under dilute aqueous acid conditions. In these hydrolysis reactions, we're going to see, for the other species, we're going to see an acid medium version and then basic reaction as well. And of course, the acidic one is more complex, relatively speaking, because we have to do all those proton transfer steps. So our first step in this is, lo and behold, a proton transfer. In this case, we do have the hydronium ion because we're trying to do a hydrolysis reaction. Then we have nucleophilic attack. We have water as the nucleophile for the hydrolysis reaction. Hopefully no surprise there. Let me correct that little glitch again. Now we need to lose methanol, so we have to do our set of proton transfers. So we use water to remove a proton. Get this intermediate here. Like so. Now we have to protonate the methoxy group so it becomes a good leaving group. So again, dilute aqueous acid conditions, the hydronium ion. What happened there? Let's try that again. Apologies for this. Okay, I'll have to live with that, but it's coming from the bond to show the proton transfer. And then we have this species here. We can do reform the carbonyl and kick out our leaving group. Now it's leaving us methanol. We have this protonated form of carboxylic acid and the methanol that we just produced, or the water. I'm going to put in the water because there's probably a lot more of that around. Not probably, there is a lot more of that around. And it will deprotonate. And we'll end up with our carboxylic acid. So that's the acid hydrolysis of the ester. We can have transesterification reactions. And the acid catalyzed one, we've already looked at the mechanism. So I'm just going to refer you back to that first video lecture, lecture of the with the PowerPoint slides, when we talked about section 20.7. That was that example acid catalyzed reaction that we looked at. Base catalyzed transesterification reaction is relatively straightforward mechanism, as you might guess. So if we were doing this with this species, so this in base catalyzed, we'd have the alkoxide of the alcohol we wanted to do the transesterification with. So we would get nucleophilic attack. Again, we just don't have all of those proton transfer steps going on. So I'm just going to shorthand that to an ethyl group, but we're switching out. We're turning in this case, I'm writing in blue here, we're turning an ethyl ester into a propyl ester. So the ethyl group becomes leaving group. I'll do the shorthand for propyl. We don't use that as often as ET for ethyl. But that's our base catalyzed, or really base promoted transesterification reaction. And this base promoted one is one that's actually used on I go back a couple of slides. On these fat molecules, if you do that with, let's change the color here, but if we do something like methoxide here, we end up generating, instead of the sodium salt to the carboxylic acid, we generate these compounds here. I'm going to switch to a blank slide. There's one here. So we can turn the fat molecule into these compounds here. And of course, the fatty acids can be saturated, like this example, or unsaturated, where we have double bonds in there. But these are compounds called fatty acid metal esters. And these fatty acid metal esters are important. If you've heard about taking used vegetable oil and turning it into a fuel called biodiesel, these are the type of compounds that you find in biodiesel, and they do it in a very similar way. But fatty acid metal esters, or FAMEs for short, they're also used in if you're doing reactions or if you're doing analysis of fatty acid composition of cells. So Dr. Champagne and I have been working on, and our colleague Dr. Jimenez from Colgate University have been working on looking at the fatty acid composition of cells and how it might change and be related to differences in metabolism. Okay. Esters will undergo, oh, one more thing before I get onto there. Esters will react with amines like ammonia to give you amides, but the reaction is not real efficient. Whoa. Typically, you would do the reaction with acid chlorides, and that's something we'll see in the next section. The last bit to talk about is the reaction of the esters with reducing reagents like lithium aluminum hydride and carbon nucleophiles like Grignards, and we've kind of seen some of this before, so we'll go through it relatively quickly. But if we take lithium aluminum hydride on our ester, it will reduce it down. We saw this actually back in Org 1. It will reduce it to the primary alcohol from the kind of carboxylic acid part, and then the byproduct will also be the alcohol from the kind of alcohol part of the ester. And just like we saw before, and we saw this in the aldehydes and ketones chapter, I should say, for this part, we can use those modified lithium aluminum hydrides where it only reacts once, and so we can reduce an ester to an aldehyde. And again, it's the bulk here of the diisobutyl aluminum hydride that is helping to mediate that reactivity. This is often done at cold temperatures too to help control it. In reality, when you're trying to do this reaction in a lab, it's a little bit tricky to control. This reaction, the Grignard reaction on the bottom of the slide here, is very similar, or not very similar. It's analogous to the reaction we talked about with the acid chloride, and it's also a reaction that we talked about in the alcohols chapter back in Org 1. So the Grignard reagent adds twice. The intermediate is a ketone. The mechanism shown in your book, I'm not going to repeat it here because it's the same kind of mechanism. No, it's the same mechanism as we saw with the acid chloride. Only instead of the chloride leaving, we have the alkoxy group leaving. Okay, so that finishes those two sections. So for some questions for you to look at, just let me flick back here. Question 2020 is a pretty good one about ways to make those esters from a carboxylic acid, and it's asking you to give 3 different ways to do it. Another good set of questions are questions 20.22, which gets you to kind of drill through the hydrolysis, some synthesis, and then the reactions of the esters with our reducing agents like lithal and the Grignards. Okay, so that finishes our section 2010 and 2011.